And now I think we move on to next speaker, who is Luc de Meester from the University of Leuven. Luc is shifting back to the present and the future maybe too. And also we probably are going to hear about smaller ponds, isn't it?
A little bit, yes. I want to thank the inviters, the organizers to invite me, because it's always a pleasure to try to make my point that evolution is not always by definition about details. Evolution tends to be ignored in all these discussions on ecological responses. I think you can learn a lot about looking at the ecology itself, but I also think that sometimes evolutionary processes can quite deeply interfere with how the ecological processes go on. And I want to make that point and illustrate it a little bit. So climate change is basically stress to organisms. It's stress not only because of the temperature, but also because of all the associated changes like eutrophication, cyanobacterial blooms, also the fact that there is exotic species coming in. So stress, that means that it's selection pressure, and this selection pressure will act both at the community level as well as at the level of the individual populations. And at the community level, it will result in species sorting, so changes in the species abundances in the communities. And at the population level, it will result in natural selection and the responsive selection, which is microevolution. And to the extent that this goes fast, there may be an interaction between those two processes, which we then call eco-evolutionary dynamics, where species sorting and natural selection will influence each other. It's easy to imagine if you have very fast evolution and species adapt to new conditions, then there is less need for species sorting. And if species sorting goes very fast and your species is immediately extinct, then of course there is no time for natural selection. So these two processes can interact. So we call that eco-evolutionary dynamics. And I think it's important because it changes our view on the ecology of species. It changes our view on the process that we are studying, because there is this potential of very high dynamics between, on the one hand, species traits changing, and on the other hand, the occurrence of the species themselves. So you should not too easily consider species to be evolutionary stable. They are constantly changing, perhaps in details, perhaps sometimes less in details. And there is quite a few reviews on this topic now. So with respect to climate change, what are the population responses to global warming? You may have acclimatization. The species may show phenotypic shifts, plasticity, so that they can stay where they are. They can also genetically adapt. They can migrate and they can get extinct. And some of them are ecological processes, and this is then the adaptation is then an evolutionary process. So we want to look into the evolutionary dynamics and climate change. And more specifically, we want to take an evolving meta-community approach, which is basically saying not more than that we are looking at community structure, we are looking at evolution, and we are looking at space. And if we say we, I have to stress that it's largely Wendy van Doorselaar who did all this work, so I'm talking perhaps more in her name, or at least presenting her work. The first thing we wanted to know is how fast is evolution? Can we actually see evolution going on? Do resident populations adapt to temperature rise in ecological time, meaning in the course of a few months or a few years? And we use the water flea Daphne as a model because it's a very convenient model, also because it has a relatively short generation time, it's easy to work with, you can do nice experiments with it. And so Wendy actually did two kinds of experiments. On the one hand, she used a temperature gradient in an aquarium setting, more or less similar to the study Brian presented this morning with the sticklebacks. You keep them in very nice conditions. They are single population studies in which you're only looking at the responses of that species. And on the other hand, Wendy also did selection experiment, the large mesocosms. These are the ones here of Brian in Liverpool. She also worked with the mesocosms of Eric Jeppesen in Neri. And so all this was part of this Euro-Olympics project. So we were exposing our populations to two different temperatures based on the IPCC scenarios. It's basically a plus 4 degrees Celsius, both in these two experiments. The basic design is actually extremely simple, but it's quite difficult to do, but on the other hand, it's simple in design. You have a selection temperature, you expose your population for a certain time to the selection temperature, then you isolate individuals from these selected tanks, you keep them in the laboratory for a number of generations to purge them from all maternal effects and so on, all the influences that they had in the tanks, so that you have sort of lines that are conditioned, that are standardized for their growing conditions, and then you test them at different temperatures to see whether they have genetic differences in their preference or in their performance at different temperatures. So the aquarium experiment, so that's basically in splendid isolation. You only have Daphnia magna in these aquaria. And just a short view on how the design was. We had 2 selection temperatures, and we had actually also 2 culling regimes. The reason why we did that is that we did not know what to do, whether we had to allow the population just to grow or whether we had to harvest them regularly. So we did both things. And we started each aquarium with 240 different clones of Daphnia magna, all coming from one population. And actually we had 5 replicate aquaria, each time with 5 different sets of 240 clones. The result is given here after 3 months of selection, so that's not long, it's 100 days of selection. We have actually a significant response. If we look at the population that we have after 3 months in the high temperature range, and we do live table experiments, then we actually see that this is a start population and this is the 2 end populations at 20 degrees and 24 degrees. And you see that the population that kept at 24 degrees has a higher population growth rate than the population that kept at 20 degrees. So we have a significant evolution, a significant change in the population growth rates in only 3 months' time. So there is a significant evolutionary change within 3 months. This is the aquarium experiment. The mesocosm experiment is, of course, quite more complicated. It's also much, much bigger. But it's more complicated because in those mesocosms you have a lot of other organisms. You have the presence of vegetation, of competitors, predators, parasites and so. And so we inoculated these mesocosms basically with the same population. It was a little bit less clones. It was only 150 clones, but it was the same population. And after 5 months of selection, we also observed an evolutionary response in those mesocosms. But the evolutionary response was not with respect to population growth. It was in this case with respect to body size. If you compare the start population with the population after selection, there is an interesting response in the sense that at 20 degrees Celsius, the animals show, if you culture them at 24 degrees Celsius, they show a reduction in body size. The animals that have been kept at 24 degrees Celsius for a few months, they actually show a much weaker response in body size. So the evolution sort of compensates for the phenotypic plasticity. So the end result of this experiment, which was quite a bit of work, is that you have rapid and significant evolution in both cases, but it's different. It's for different traits. And it was also difficult to predict the response in mesocosms. In the aquaria, we had predicted that it would be population growth rate, and we observed population growth. In mesocosms, we had also predicted that it would be population growth rate, and we observed something completely different. This is not so surprising because the conditions are so complex, and you have predators, you have parasites and stuff. So the different ecological setting yields a widely different evolutionary response. But in both cases, we did observe rapid evolution. Evolution within a few months, you can at least say that this is within ecological time, that is within one growing season. So what? We do find rapid evolution, but does it actually make a difference? Does it actually impact the system? So that's the second question we wanted to ask. So whether genetic adaptation actually has a consequence on those populations or community processes. So to introduce you to that experiment, I would like to indeed introduce you to climate change from an evolving meta-community perspective, which I think is a very important perspective to think about. If you have a local community somewhere in Liverpool exposed to climate change, then there is a number of different responses that you can expect, and those will interact. First of all, you have local processes. It's the local community that can show shifts in species composition. You may have the local population that can show responses to natural selection, so changes in trait values. And importantly, these two things may interact. That's the slide I started my talk with. But there is actually more, because you may also have immigration. You may have immigration of species coming from the south, or from warmer regions, or you may have immigration of new genotypes from the same species of resident species already. Again, these two things may interact. And more importantly, probably, is that these two processes, again, may interact strongly. So you have actually 4 interactions. You have interactions between species sorting and natural selection, and at the same time you have also interaction between local processes and regional processes. The processes that occur in your lake and the processes that occur by migration. So what we have done so far, and those were quite large experiments, is actually only showing that there was genetic evolution, that there was genetic changes through time. So the only thing so far we have shown is that there is microevolution in populations. We still need to do all this other stuff. So we did not do everything. We failed actually for some parts of it, but at least we did one thing extra, is that we wanted to see whether there would be an interaction between the microevolution and the immigration of new genotypes. So actually what we did is we went to the mesocosms, and in the warm water mesocosms we did competition experiments with local resident clones from the UK and clones we brought from the Camargue area in southern France, and we did a competition experiment between these two genotypes, trying to find out actually whether if you have genetic adaptation to a warmer climate, whether that would actually reduce the impact of immigration from warmer areas, whether that would reduce the success of immigrants. So this is the experimental design, so this is what it looks like. We had our UK clones, we had French clones, we kept them in 3 different ratios. The first ratio is the 1-1, you just test fitness, you test which clone is the best. And we also had some more realistic ratios, so 1-10. I mean, the idea is that the French clone would arrive in the UK population, but of course it would be at lower frequencies. Of course, this is still far from realistic, but it's not easy to do an experiment where you have one individual in 10 million locals. So that we could not do. We had 2 non-heated mesocosm populations and 1 heated, which is an adapted to warm temperature, because all these experiments were done in a warm mesocosm. We only had 1 heated population. That's something that is worth mentioning, because even though I've been claiming that there was rapid evolutionary adaptation to temperature change, we also have to admit that after 2 years, most of our Daphnia magna populations were extinct in the mesocosms. And the reason was that there was a massive development of plants. So even though those water fleas could be very proud that they were adapting to the high water temperature, they actually all got extinct. In the transplant experiment, the first result was that if we put French clones and UK clones in warm conditions for 35 days, which is not so long, it's 1 month, then we see that after a month we can't find back the UK clones. Everything is dominated by the French clones. We could determine the relative abundances by genetic markers, so we knew what the genetic markers were that were associated with the French clones. So actually after 1 month, everything we find back are French individuals. And that's also when they were inoculated at much lower densities than the UK clones. So the French are massively better than the UK clones at high temperatures. So the French ones are invading UK populations at a massive rate, if they would be brought in by birds or something like that. But look what happens if we allow the population to adapt for 1.5 years at higher temperatures. Then we see that there is a major reduction in the dominance of the French clones. I have to be honest with you, this is the line of equal fitness, so the French clones are still better than the UK clones. They are still outcompeting the UK clones. But it is much less strong dominance. And at lower densities you see that actually you can find many UK clones back. So basically, evolution here changes the relative importance of regional versus local factors. If you have immigration, the immigrants will have much less success if you allow also evolution to happen. And it's evolution at a very fast rate. It's evolution during the course of a few months. Actually, Joost van Overbeek, a postdoc in our group, has modeled this. He has fitted a Lotka-Volterra competition model to this system, and he can predict, so he could extrapolate, and what would happen if you would have, for instance, 1 French individual invading a population of 1 billion UK individuals. And then still, we see actually that the French always win. So under this 1.5 years of evolution, the French would still invade the UK. But it does make a big difference whether or not the UK clones are adapted. And I think this graph shows it the nicest. This is just, it's perhaps This is the ratio of immigrants, of French clones to immigrants. So these are sort of normal ratios, you could say. 1 in 100 million or 1 of a billion individuals would be French. And then you have to imagine that the French will always in the end win over the UK clones. But here it is indicated how many more French clones you would need to have an equal number of French and UK individuals at the same time. And so that would, if you do the calculations, that turns out to be something like 10,000 or 100,000 times more. You need many more immigrants to have the same effect if the UK clones are adapting to the temperature change. So basically, the conclusions here are that there is rapid and also adaptive evolution. We can show that it's adaptive because actually they become better when you are competing them against French immigrants. So it's really adaptive evolution. It makes a difference. And I hope I have also shown or could stress the importance of the evolving meta-community context, thinking not only in terms of species, but also evolutionary rates, and also the fact that you have local and regional processes. And I think it's very important to realize that models that ignore evolution in predicting the outcomes of climate change will actually sometimes make big mistakes because, for instance, if you have also resident population, they will probably overestimate migration rates compared to local processes. This is an overview of some papers that all have rather more ideas and review papers on evolutionary aspects of climate change. There is actually not so many data around. I would also like to stress that even though I'm making a plea now to look into evolutionary dynamics and the impact on ecological dynamics, you should not think that evolution will solve our problem and evolution will sort of make climate change worse, less bad. Because the claim I want to make is that evolution will result in different responses, but not necessarily less strong responses. It will change the dynamics. It will not make the dynamics completely irrelevant. I would like to broaden the perspective a little bit with a few statements. First of all, what I did so far is showing you that evolution may localize processes at the population level. The question may arise whether it would also impact communities and ecosystems. And here I have to say that our experiment failed. We tried to also do an experiment on the impact on communities, but our communities that we used in the Liverpool microcosms in the end turned out to be so variable from the start that our data set was going in all directions and we could not find a signal. The idea is, of course, that you would, of course, also look at species sorting locally and the immigration of new species, and you would do all these experiments. Because I've now outlined quite a few experiments, but we still only are discovering part of the process. Just to illustrate the idea, think about climate change imposing on a beautiful community of zooplankton. You may have a shift, for instance, towards smaller species or something like that. And on the other hand, it may be that the Daphnia can adapt genetically to cope with the climate change. And as a result of that, you may have still Daphnia in the population. You all know, because you're all familiar with freshwater systems, that this may, in principle, result in the difference between clear water and turbid states. I'm not saying that it's evolution that does the job. I'm saying that under some circumstances, it might be that evolution helps keeping you in a clear water state. And actually, our experiment in the UK failed. But Cathy Duvivier did another experiment in which she actually could show such a community impact of adaptation. The only difference is that she did adaptation to fish predation pressure and not so much to climate. But the basic idea is the same. She exposed populations to fish predation pressure, and then she worked with adapted versus non-adapted populations, and she put in those containers here a whole lot of individuals of zooplankton from all kinds of different ponds, and she looked at what community she would get. And the idea was that if you have adapted Daphnia populations, that Daphnia would actually be stronger in competing with the other species than if you have non-adapted. And this is the result. Actually, the results are quite variable again, but at least the general trend is very convincing. These are the adapted populations, and these are the non-adapted populations. This start has moved because it should be here. The non-adapted ones are the start populations, so the population which we started to inoculate the containers. This is the adapted ones after about 1 year of genetic adaptation to predation pressure. And the orange pieces of the cake here, this is the abundance, the relative abundance of Daphnia magna in these communities. And you see that in the left pies here, you have a much stronger impact of Daphnia magna than in the right ones, which means that Daphnia is much more important in that community when it is adapted than when it's not adapted. So actually, the fact that you genetically adapt your population to the conditions will make it stronger in competing with other species. I have to admit that Daphnia magna is a very strong competitor, so you will not have this with all species. And can it have ecosystem impacts? We didn't do the experiments with Daphnia yet, but at least there is recently a publication in Nature by Harman et al. Working with two different kinds of sticklebacks, belonging to the same species, but two different feeding morphs. And they used mesocosms to inoculate them with those different sticklebacks. And they looked at a number of ecosystem characteristics, like the amount of dissolved organic carbon and so. And they could find significant differences, whether it was the limnitic versus the benthic sticklebacks that were added. So just adding different genetic lineages of fish into mesocosms resulted in different ecosystem characteristics. So yes, also genetic differences in species can have impact at the ecosystem functioning. The second thing that I would like to stress actually is climate change is much more than temperature. I mean, all the lectures this morning were about that topic, and I think it cannot be stressed enough. And also, if we then think about evolutionary responses, I think this is a very important point. You have, of course, the classic thing of eutrophication and the associated cyanobacteria blooms. Cyanobacteria blooms, they produce toxins, so that is a very strong selection pressure on zooplankton. Also, the occurrence of exotic species. Exotic species change ecosystems. They are often occurring at high abundances. It's a novel environment, so that is strong selection pressures. So in each of these cases, I think, you have the potential of having strong eco-evolutionary dynamics. And space is very important there too, especially also in the case of exotic species, which puts you, again, in this framework of having communities, evolution, and space. You may think that what I'm saying is only relevant for Daphnia, and Daphnia is a bit of a weird organism perhaps, but I would like to sort of stress that this is unlikely to be the case. And the basic idea is that you have an environmental change, you have genetic adaptation to it, and it's the conflict between adaptation and immigration that actually will determine what is going on, also with respect to climate change. And if you're then looking at this interaction between adaptation and immigration, I think you can make a sort of guess about what body sizes will do. And then Daphnia is about here, and you have dispersal rates, population growth rates, and rates of adaptation that will change with body size. And judging from this sort of generalization, you can imagine that at the bacterial level, at the protist level, at algae and so, you will have much more of these eco-evolutionary dynamics even than for Daphnia. But even at the level of fish and sticklebacks, there is more and more reports of quite short-term changes in genetic structure, short-term adaptation to different regimes. So I would not ignore the impact of evolution in those vertebrates neither. It may take a few years rather than a few months, but it's better not to ignore these things. And so what I would like to claim here is that the impact of eco-evolutionary dynamics also on your water system is probably quite important, and especially if the system is changing very fast, because that creates new environments and new selection pressures. So if you get cyanobacteria blooms, if you get exotic species, or if you have state shifts and so, if you do severe management impacts, so that you, for instance, clean up a lake, this is also a major selection factor on all kinds of organisms. And I just wanted to close by giving you some slides on what we are doing now, on ongoing research, which is actually done by Aurora Geertz, who is there. Because we are now applying resurrection ecology on this problem of climate change. As Daphnia produces dormant eggs that are sort of archived in the sediment, you can take sediment cores, you can hatch recent population and old population, and you can compare them. What we want to do is to do that with a number of replicate sediment cores, part of them delivered actually by the lab of Rick, and which we will isolate animals from 1965 and 2005 and compare therefore their responses to temperature. So we will actually revive flower power Daphnia, and we will look on how the responses are to temperature. And by looking at different replicate sediment cores, we are hoping that we can extract the signal of temperature rather than all the other disturbing signals of, for instance, a change in fish predation pressure or eutrophication. And so that's Aura Geertz who is going to do that. And Louisa Orsini is also involved because she is going to apply paleogenomics on these questions. She's going to apply a whole battery of genetic markers on the Daphnia from the different layers. And we hope, so by doing it together with Aurora, Aurora will look at traits, Louisa will look at the genetic markers. By linking the traits to the genetic markers, if we find the markers, and if you find the genes that are involved, then this opens very nice perspectives because with the DNA, you can go much further back into time. And we would like to, for instance, go to the Middle Ages where we have that short ice age, or perhaps to prehistorical times. If you could do that, that would be really fun. And I want to end by showing you, or thanking the local team for all the work they invested in all these different questions. And as it is always in evolving meta-communities, there is also a regional impact. And I really want to thank both Brian and Eric for the opportunities they gave us to work with our mesocosms, and of course Rick also for leading the Eurolympics program, which is one of the funding sources of the work I've been presenting. Thank you.
Thank you for this very interesting lecture, giving a new perspective. I especially like the term resurrection ecology. It's quite an interesting thought.
I did not invent it. So.
It is maybe interesting to see what kind of organisms we can revive from the past. It's, whole movies have been made about that, right? Yeah. So there is time for a couple of questions. Who can I give the floor? Yeah, please, you go.
In this particular case, we don't have. We have done another reconstruction using a resurrection ecology. We did a reconstruction of the response to predation. And in this case, we saw that there was very rapid evolution towards being better defended against fish in a few years. But we also saw when the fish disappeared, so later on in the core, these traits also disappeared. So really showing that there is a cost to these traits. So you have a very dynamic situation. I think it's whether you have a selection pressure in one direction or the other, it doesn't, well, everything has costs basically.
Questions? Yeah, Martin.
Yeah. Yep. I think that it's a very good question. I think.
Can you repeat the question very
Oh, yeah, sorry. Whether it's the fact that Daphnia has this alteration of clonal propagation and sexual reproduction makes a difference. Because it's indeed a very powerful reproduction mode in the sense you can grow very fast, you can have very nice selection among clones, which is very effective selection, and then you generate from time to time genetic variation. So it has been claimed that evolution goes faster in those organisms that do have those systems. I have two answers to that. Yeah, three actually. The first one is yes, it makes a difference. The second one is that there is a lot of organisms around that do the same thing, like algae, protists, they all mix sex and asexual reproduction. Plants do. So there is actually a whole lot of organisms that mix those two strategies. So it's probably more abundant than pure sexuality. And third, also in purely sexual organisms, like for instance in the Darwin finches, they have reported changes in the beak morphology with time over the years. So during the course of a few years, you also find these in vertebrates. So the answer would be yes, it makes a difference, but also in vertebrates you find it at the order of a few years.
Thank you. I think we are going to move on. Thank you for this very interesting talk about rapid evolution. You never think about it, that it happens in such a large degree.